So this begins at the end, towards the end of part 1 of the blood. This is actually before hemostasis that I covered today in lecture. So we're going to be talking about leukocytes, white blood cell, and the function that they play in the immune system. Okay, so leukocytes, also called white blood cells, are much larger than erythrocytes or red blood cells, and they have a very prominent nucleus. So when you're looking at a histograph, the nucleus will appear a dark purple or blue. They are found in much less number or frequency than red blood cells, and the biggest reason is because they use the blood as transportation, basically highways to get to where they need to go. Usually they don't function in the blood. They will leave the blood, enter tissue, where they will carry out their functions. So when they basically get to where they need to go, the tissue that may potentially have some type of pathogen, they stick or adhere to the edge of the walls, and then they squeeze between the tiny spaces in the endothelial cells. We call that immigration. And they know where to go because it's that chemotactic event that occurs when tissue gets injured. Leukocytes, or white blood cells, are divided into two basic categories. Granulocytes, which have visible cytoplasmic granules, and agranulocytes, which means they lack visible. They still have granules, you just can't see them. Okay, so first we're going to talk about the different types of granulocytes. The nucleus will be a lot of times what we call multi-lobed. So it kind of looks like it might have more than one nucleus. But if you were able to get it in the right section, you would be able to see that it has all these connecting pieces. Okay, so a lot of times they have multiple lobes like that. The multi-lobed is more what you'll see in the neutrophil. Bilobed is going to be a characteristic of what we call an eosinophil. The cells will all have some type of granule that will basically appear over. Sometimes they can kind of cover the nuclei. So they're within the cell. And usually we actually distinguish the type of leukocyte based on what color the granule happens to be. So basophils pick up a basic stain, which is blue in color, methylene blue. Eosinophils pick up an acidic stain or dye, which is more red or orange. And then neutrophils, which are actually the most common out of all the leukocytes, about 50 to 70%, actually absorb a little bit of both. So they don't have a super distinguishable color. It's more of a really light purple. You can kind of tell stuff's there, but it's not red and it's not blue. Neutrophils, again, the most common, they are very important phagocytes in the body. So phagocytosis is when you engulf and destroy basically cellular debris or any pathogen. They're really important specifically against bacterial cells. You can also call neutrophils polymorphonucleocytes. On the exam, however, I will use neutrophil. Why they have that is because they have 3 to 5 lobes. Younger ones will have 3. The more mature they get, the more lobes they will have. So again, during injury, hemostasis is triggered. Also, the chemicals that are released will attract neutrophils. They're one of the first cells that will arrive on scene. And again, we call that chemotaxis. So it sets off an alarm. So the neutrophils will adhere to the edge of the wall. They'll squeeze out, immigration, and they're going to go towards that damaged tissue. So neutrophils can directly kill bacterial cells by engulfing them, and they basically increase the chemotactic event, and that more neutrophils are going to come to the area, other white blood cells that might aid, which also increases the inflammation process. Remember, inflammation is the first step in tissue repair. It's also an important part of immune function. Immature neutrophils are called band cells because if you look at the nucleus, it kind of looks like a curved band, C. If you have an increased number of these, that basically means that your body is making more neutrophils than it normally would. And so some of these are released when they're still slightly mature. We call that a shift to the left. So again, when you have an increased amount of bacteria, most commonly an infection, which can easily follow a viral infection. Eosinophils, again, pick up that eosin red-orange stain, typically have a bilobe nucleus. They are also phagocytes. They are most important in parasitic worms, and they're also going to increase in number during allergic reactions. So the granules in their cytoplasm contain enzymes and toxins specific to different types of parasitic worms, and because of the allergic reactions, they'll also control and induce inflammation. Basophils are the least common leukocyte. Never let monkeys eat bananas, which is your frequency of white blood cells. So again, neutrophils are the most common, then lymphocytes, then monocytes, eosinophils. So again, basophils are the least common, usually about 0.5% in the body. So a lot less than about the 50 to 70 that neutrophils are. They have an S-shaped nucleus, although a lot of times you cannot see it because the granules that are very darkly stained will cover it. The granules, though, contain two very important chemicals, one of which, histamine, helps mediate inflammation. The other is heparin, which, as we talked about earlier today in lecture, heparin is an anticoagulant or blood thinner. Okay, so what they actually look like, you should need to know this for your lab test tomorrow. So again, neutrophils, if you look at the granules, they don't pick up a super distinguishable stain. Eosinophils have some redness to them, and you also see that bilobe nucleus. And then the basophils, again, have the very dark staining granules that are kind of surrounding or covering the nucleus. Okay, the other basic class are agranulocytes. So they have granules, you just can't see them. Two types, we have lymphocytes, which are the second most common. Lymphocytes look very different than the others. They have the largest nucleus, very little cytoplasm. They function very different than any of the other white blood cells or leukocytes, and that they are highly specific against the pathogen that they will try to get rid of. Two different types of lymphocytes, we'll discuss that when we do the lymphatic system. Both, again, are highly specific by which antigens basically stimulate them. Remember, antigens are highly specific cell markers. So the two types of lymphocytes, we have B lymphocytes, also called B cells. We will talk in a lot of detail how these become active in immune system, but for right now, basic functions, I would like you to know that they make or produce antibodies. And as we talked about with blood typing, antibodies can bind to the matching antigen and they mark a cell for destruction. Again, they're highly specific, so each B lymphocyte basically responds to its own antigen. The other type of lymphocyte are T cells or T lymphocytes. Again, it's specific. All lymphocytes are highly specific. These do not make antibodies. They can directly kill and destroy the appropriate, very specific cell. So again, antibodies and the T cells are very, very unique. And again, we will discuss that more with the immune system. The other type of agranulocyte is a monocyte, which is actually the largest of the leukocytes. Usually has a indentation in the nucleus, kind of U-shaped. We call it a monocyte when it's in the blood, or we call circulation. As soon as it enters tissues, we now call it a macrophage. A macrophage is phagocytic. Macro is large. It actually has the ability to ingest and break down cells and debris that are larger than it itself. It activates other components of the immune system by basically showing these foreign pieces that we want to attack. It's very important in activating lymphocytes. Again, we'll talk about that more with immune. So again, the lymphocyte is going to have a very large nucleus, very, very little amount of cytoplasm around it. Monocyte is the largest, and actually if you look at the lymphocyte here, it's the smallest, other than the red blood cells, smallest of the white. Monocytes are the largest of the white blood cells, U-shaped nucleus, really large. So why sometimes would we need to know why, for instance, neutrophils are the most common white blood cell? This comes into play whenever you are you're looking at blood to see the health of an individual, or we call a complete blood count. This is a test for anemia, possible not enough red blood cells. We talked about a few different types previously and other conditions that we'll discuss now. So blood sample can be taken, it will be stained. You can look at it under a microscope, or most days now they have basically a machine that does this for you. And essentially how it works, if this is your slide of blood, they're going to really quickly make sure that they survey and do this order, counting the cells. Usually they count 100, and out of 100 cells, or 1,000, how many of each type do they have? Red blood cell counts, they do cells per milliliter. They use that to calculate the hematocrit, or hematocrit are the number of red blood cells that you have. And of course, the main component of red blood cells is the hemoglobin. So by getting the hematocrit, you can get the hemoglobin concentration as well. Red blood cells, you're looking at the cell size, the cell volume. You want them to be nice, plump, healthy. You don't want them to have a sickle shape so they can carry more hemoglobin. For platelets, you look at how many and the space that they take up. For white blood cells, you look at the total number of leukocytes, and then you can also look at each individual one, the percentage. So this is basically what a normal healthy individual should have. Okay, so erythrocytes, you have around 5 million. Okay, platelets, about 150 to 400,000. And leukocytes, again, this is just in blood, 5,000 to 10,000. Again, this is per millimeter cubed. Or in other words, per milliliter. Neutrophils, roughly, you don't need to know these exactly, but neutrophils, depending on the book, 60 to 70%. Lymphocytes, about 20. Monocytes, typically around 5. Eosinophils, less than 4. And basophils, less than 1. So very, very low accounts. And again, this is in blood, not necessarily in the body. Remember, when you look at this, we did the whole blood, the buffy coat, which is less than 1%, is going to contain your platelets and leukocytes. The rest of the formed elements, making roughly 44%, are going to be all of your red blood cells. So especially when we are looking at white blood cells, a few different disorders that you can have associated with those. Leukocytosis is when the white blood cell count is greater than 10,000 millimeter cubed. This typically indicates that a person has a bad infection, and that's why they have so many more white blood cells to help fight it, or potentially they could have cancer of a particular white blood cell or the bone marrow. Leukopenia, on the other hand, is a white blood cell count that's going to be lower than normal. Anything less than 5,000 usually indicates some type of disease in which the body is unable to produce enough white blood cells to protect itself. So patients will get this during AIDS. We'll talk a lot more about that with immune, if the bone marrow is not producing cells properly, if a person is really malnutritioned, or chemotherapy, which kills cancer, also kills a lot of bone marrow cells, including leukocytes. So we had hemopoiesis or hematopoiesis, which was the process of making all blood cells. You had erythropoiesis, which was making red blood cells. And now we have leukopoiesis, which is the process of making white blood cells. All cells are made in the bone marrow. So leukopoiesis, you have hematopoietic stem cells again, just like with red blood cells, and they start to divide, differentiate to eventually become a specific type of formed element, whether it's going to be a red blood cell, a platelet, or any of the 5 different types of white blood cells. So hematopoietic stem cell will produce 2 different cell lines that essentially all other cells arise from. So the myeloid cell line produces almost all the formed elements, including erythrocytes, platelets, and all of the white blood cells other than lymphocytes. The lymphoid cell line produces lymphocytes that are derived from immature cells called lymphoblasts. So the myeloid cell line, the cells continue to differentiate into blast cells. The blast cells will then become a monocyte or any of the 3 types of granulocytes if it's going to be a white blood cell. Of course, if it's going to be a red blood cell, they will start to differentiate into that immature red blood cell. The lymphoid stem cell, again, will create the B and T lymphocytes. And we actually name the B and T lymphocytes based on where these cells go to mature. So B lymphocytes stay in the bone marrow for maturation, hence B. T lymphocytes move to the thymus where they mature, which is why they're called T. Okay, so again, if you're looking at the commonalities, all blood cells originate, or formed elements originate from hematopoietic stem cell. The myeloid stem cell is going to differentiate everything except lymphocytes. So lymphocytes will differentiate the lymphoid stem cells. Lymphoblasts will then have mature T lymphocytes and B lymphocytes. The myeloid stem cell, and again right now we're focusing on leukopoiesis only, so the formation of white blood cells, either becomes eventually a monocyte, or it can become any of the 3 granulocytes that we have. So 4 out of the 5 white blood cells are derived from the myeloid cell line. Leukemia is cancer of a blood cell or the bone marrow itself. There are different types of leukemia depending on the particular cell. So we have acute or chronic based on how fast the disease is progressing, the severity of it. So acute happens very rapidly. You can tell it's occurring because the body will actually start releasing a lot more of the immature cells into the blood. Chronic, it's much slower to release these immature cells, so it takes longer to typically diagnose. They're also classified by which cell line do the abnormal cells come from. So lymphocytic, of course, come from the lymphoid stem cell line, generally the B lymphocytes. Those are the ones that stay in the bone marrow. And then myelogenous come from the myeloid stem cell. So any of the myeloid cells, again, that's going to be your monocytes, the eosinophils, your basophils, and the neutrophils. So acute lymphocytic leukemia is the most common that you have in children. So it's going to occur very rapidly. Lymphocytic, it involves the lymphocytes. So, unfortunately, not only do you have this increased number of lymphocytes in the blood, but it crowds the bone marrow. The bone marrow is focused on producing those cells. So, you do not have as many of all the other cells, red blood cells, or any of the other white blood cells. In fact, because it gets so crowded, a lot of times cancer then spreads to other tissues. And we use the term metastasize when this happens. So, how they would treat or diagnose the leukemia, usually, again, acute, will metastasize really quickly. It's maturing very rapidly and require a much aggressive treatment, typically chemotherapy, radiation therapy. It may even involve amputation in some cases. So if we do some review questions, which of the following statements is false? Hematocrit is the volume by percent of our blood cells found in the blood. That is true. Males are going to have a higher hematocrit than females because of testosterone. Band neutrophils are immature cells. That's true. When someone has a bacterial infection, the number of these will increase. A monocyte is the type of white blood cell. True. They're called macrophages in tissues. Anemia is when there's an abnormally low number of red blood cells or erythrocytes. That's true. So, of course, the incomplete statement are red blood cells, white blood cells, and platelets are whole cells found in blood. What makes that false are platelets. Platelets are fragments of cells. They come from fragments which we call monokaryocytes. An increase in white blood cell count above normal levels is called leukocytosis. Leukopenia is a low amount. Erythrocytosis has to do with red blood cells. Immigration has to do with the squeezing of a white blood cell from a blood vessel into tissue. And erythropoiesis, of course, red blood cells, the production. Which of the following is not a factor in destroying and attacking pathogens? Chemotaxis, that's the alarm that goes off that lets white blood cells know where they need to go. The release of enzymes occurs by several white blood cells in the area. That's how they can start performing their functions. Erythropoiesis, red blood cells have to do with carrying gases, oxygen and carbon dioxide. Has nothing to do with pathogens. Immigration is the movement of a white blood cell into a tissue. And again, it knows how to do that from chemotaxis, so that's true. And phagocytosis is a capability that several white blood cells have. The two major phagocytes are the neutrophils and the monocytes or macrophages in tissues. Which of the following are the most numerous white blood cell? That's never let monkeys eat bananas. So, of course, N for never is neutrophils. Typically, they're 60 to 70%. Okay, now we're going to discuss platelets a little bit. Today in class, we did talk about hemostasis. However, we did not specifically talk about platelets or where they come from. So we're briefly going to do that now. So again, platelets are the only formed element that is not a whole cell. They, again, are cell fragments. They're the smallest of the formed elements. Their main function is in hemostasis, which of course is the process of stopping blood. So because platelets are a fragment, they do not have nuclei or other organelles found in most whole cells. Keep in mind, that's a similar characteristics of red blood cells. Mature red blood cells don't have nuclei or very many organelles either. Platelets have granules, basically just like white blood cells. However, theirs contain clotting factors, which are necessary in hemostasis. Several enzymes that are also important in this process, mitochondria for energy, glycogen, which of course is necessary for cellular respiration as well. Therefore, they can carry out oxidative catabolism. So they're going to be breaking down by losing an electron. That's what oxidation is. They also have cytoskeleton components, actin and myosin. And as we discussed with hemostasis, this is important when they contract and bringing the vessels together and sealing off the wound. Okay, so platelets themselves, fragments of cells, again, some mitochondria, they really have a lot of actin and myosin components to them, and then the granules that contain any of the 12, the different clotting factors, Roman numerals 1 through 12. So poiesis, again, is the formation of. Thrombo is another name. Thrombocyte is another name for platelets. So thrombopoiesis is the process of making platelets. Just like all of the other cells, it comes from that hematopoietic stem cell. And now it's going to differentiate into a megakaryoblast. These mature, become megakaryocytes. They will undergo mitosis, so they're making several copies of themselves, but they're not dividing the cytoplasm. So mitosis is a type of nuclear division. So basically each of these nuclei are basically forming the different platelets. Okay, mature megakaryocytes when stimulated by hormones. So we have thrombopoietin. Thrombopoietin stimulates the production of platelets. Remember, erythropoietin will stimulate the production of red blood cells. So you have cytoplasmic extensions that will leave the bone marrow. They go in the sinusoids. They break off into tons and tons of these little platelets off of the megakaryocyte. Platelets, because they're involved in hemostasis, have a very short lifespan, only about a week or maybe a little over, before they're removed from circulation by the liver and the spleen. And again, that's to keep the quality of the enzymes that are in the granules and the clotting factors that they contain. Platelets or thrombocytes are made from the liver and in the kidneys. And they basically control how many platelets are in circulation. You want to keep about 150 to 400,000. 400 is obviously going to be a little bit higher if you have been injured. If you're working out a lot, you would expect that. Okay, so thrombopoiesis, again, you have the hematopoietic stem cell, still the myeloid cell line. Again, everything's the myeloid cell line except the lymphocytes. Differentiates into a megakaryoblast. It has little arms of cytoplasm, and tiny little pieces of that start breaking off, and those are what the platelets are. So the substance that stimulates the formation of platelets, that is a hormone, thrombopoietin. Immunoglobulins are antibodies. That's the same thing. Erythropoietin stimulates the production of red blood cells. Cytokines are chemicals that are important with white blood cells.